Hello, my name is Alan Young and I'm privileged to be interviewing world-famous psychopharmacologists to tell us about their views about psychopharmacology and more particularly about the British Association for Psychopharmacology. So, Professor Trevor Robbins, if we can start perhaps by you briefly telling us a little bit about yourself and what you currently do.
Yeah, I'm currently Professor of Cognitive Neuroscience and Experimental Psychology at Cambridge. I'm the head of department for the Psychology department there, and I direct the Wellcome Trust MRC Behavioural and Clinical Neuroscience Institute.
And you're one of the most cited scientists in the whole of medicine, which you very modestly didn't say yourself, because your impact in the field has been enormous.
Well, thank you for saying that. I think the impact has to be measured a number of ways, of course. I've been very happy to have excellent collaborators.
If we can just go back a bit, Trevor, and talk about how you came into the field of neuroscience and psychopharmacology, perhaps you could help us understand how you got to where you are today.
Sure, yeah, I was always headed for science. I was turned on by the genetic code, having read about it on the Evening Standard, actually, on the top of a bus, and I escaped from South London to Cambridge in the late 60s, and because at Cambridge you can do a number of subjects, and I ended up doing psychology as well as biochemistry in the second year, and I met Susan Iverson, and she turned me on to that interest interface, which I guess I've kind of prosecuted by specialising ultimately in psychology. I was going to do clinical psychology at some point, did too well in the exam, and then stayed on to a PhD with Sue and also Les Iverson, who influenced me early on, and never looked back from there. I mean, fantastic excitement in neuroscience and bringing everything together with psychology.
So I guess even that brief resume shows some of the real attractions of psychopharmacology, because it's people from perhaps different backgrounds, from psychology, from pharmacology. There's a possibility of working with world-class minds, and it's a very, very exciting field. Would all that be true to say? Absolutely.
I mean, it's the interface, isn't it? Psychiatry, I think, is such a fascinating area. Helping people, of course, is important. Neuroscience is explosive. Psychology, you know, part of the tools, the conceptual tools for exploring both of these other areas. So it's the interface that's all important, and psychopharmacology is a great exemplar of that, and a very pragmatic one, because you're actually trying to do something for people to make them better.
And I think you continue to work with colleagues within the broad field of psychopharmacology from a variety of different backgrounds, from really quite basic scientists through to clinicians.
That's right, yeah. I mean, early on I worked mainly with experimental animals. I was fascinated by the dopamine system and amphetamine and all of that. But I always had that interest in the clinical side, and we also expanded our work enormously to use new techniques, explore other species as well. So gradually I got very interested in this notion of translation, of making animal work really relevant to clinical work, justifying the animal work even more so, and also enhancing the animal work by top-down work from clinical studies, so that the bridge is strengthened. I still think a lot has to be done in that way.
Sure. Yes, I was struck a little while ago. There was an article on the BBC website, the BBC News, about a tablet that would be given to GPs to screen for dementia, and it was one of your neuropsychological tests that was going to be used for this.
Sure, yeah. So the infamous Cantab battery, which is a set of cognitive tests inspired in part by what we know about animal cognition, but also turning human tests into tests that other animals can do.
So going both ways.
Both ways. Yes. And one of these tests is a object and location memory test, and the hippocampus, the brain, which as you know is very much vulnerable in Alzheimer's disease, it's hurt early on, actually binds together objects and locations. That's one of its main functions. And so clearly a test which really gets at that particular process is very important. And it turned out that this object location task can predict Alzheimer's disease, I think nearly 3 years before formal diagnosis in a group of MCI patients. So it's been tough convincing everyone that it's going to be better than a blood test or imaging, but I think the real data still have to be collected. But I wouldn't be surprised if it's at least a much less expensive option than imaging and blood tests, probably.
Sure, but that's a great example of translation. So it's from the dreaming spires of Cambridge to your GP's clinic, really.
Yeah, I think it's a bit of fun, isn't it?
Yeah,
you know,
absolutely.
a test which just 10 minutes worth of fiddling around on an iPad. And, you know, iPads didn't exist in 1988 when we invented that test. They were very clunky things that you, touch screens that you put on your front of your computer monitor, but now it's all been...
I remember I had one.
But we're able to use this fabulous technology now.
So, if we can just focus on the BAP, I mean, you've been a very senior figure in the BAP over, well, I don't mean to say all of its 40 years, but of a considerable period of its history. So perhaps if we could, if we could hear a little bit about how you became involved with the BAP.
Yeah, let me just, so my first memory of BAP, because Sue Iveson again was someone who introduced me to it, I think I gave a talk at a BAP meeting at Churchill College, Cambridge, circa 1976 it must have been, something like that.
So that's pretty early on.
That was pretty early on. And then again at Birmingham in 78, I was in an ADHD symposium where we debated whether Ritalin really produces its effects in ADHD via paradoxical actions or other actions. And then I didn't have too much to do with it for another decade, apart from the odd meeting, when I was suddenly recruited onto council. And eventually working my way up through the various jobs you do for BAP, I became president around 96, which I enjoyed very much. It was great. And so, yeah, and I've stayed with BAP all along. I think I must have been to half of its meetings, probably. I miss some of the workshops they used to have in London. You know, those used to be great as well. And those volumes that came out were quite influential at the time.
Yeah, I think it's fascinating that such a senior figure as yourself had early positive experience presenting at the BAP, because that's really what we believe. We believe that the young people presenting at this meeting today will be the future Trevor Robbins and so on, you know, 20 years' time perhaps.
Well, I think these prizes we give are very important in a way because they highlight the excellence of the young people we have in this area. And I think also it gives them some optimism that they're actually working in an area which is good and is going to be worthwhile in the future.
So if we can just turn to the future, I wonder if we could perhaps deal with the sort of general future of the field and then perhaps move on to the BAP. So if I can ask you about the general
so one has to be a bit worried about the attitude of pharmaceutical companies, although many more are still in the game than one realises actually.
Indeed,
and I think they're monitoring carefully, and all sorts of interesting lines like the psychopharmacology, which you know is one new area. I think what's going to be important in the future is much more refined diagnoses, and there's going to be a nosological revolution in psychiatry, and we're going to be measuring things more objectively. I think that will help. I think there's going to be a lot of repurposing. I mean, we've targeted a lot of the mechanisms. We know the genetic approach is exciting, but very often, you know, after they've done the systems biology, it all converges on a particular receptor that we already know about. And we probably have many of the tools that are going to be useful for interrogating those receptors in future. So I think we have to do a bit of classical serendipitous psychopharmacology like we used to do to try and work out which disorders respond best to, you know, to some of the mechanisms we already have. So it's obviously a period of retrenchment and so forth, but I think it could be still very optimistic for the future.
Yes, I mean, I guess one thing that strikes me is that technology is advancing. We spoke about the iPad app, which you were instrumental in developing, but also technology from brain scanners to ways of monitoring, to everything in the lab.
Mobile phones, you know, for monitoring patient status, brain scanners, obviously with fantastic powers of resolution, just genetic sequencing, so stratifying patient samples, pharmacogenetics is clearly going to be important in the future because we know from some of our own experimental studies that you can greatly enhance the response to a drug like sulpiride, even in, you know, healthy volunteers, depending on whether they have the TAC1A polymorphism of the D2 receptor. So I mean, you know, the complexity obviously will be mind-boggling, but I imagine sooner or later you'll be able to more or less predict drug effects in widely variable populations.
Yes, I guess one of the issues in psychopharmacology has always been the complexity of the data, but computing processing power is going up so much that this should be solvable in the future.
Absolutely. One of the things which excites me though a lot is the molecular biology. So DREADS, designer receptors exclusively activated by designer drugs. And this is really exciting. You have these receptors, you can put them into a particular brain area, you can express them from one of its projection zones only with this Cray recombinase. This is in experimental animals, of course. And then you can give an otherwise inert ligand, which turns out to be clozapine N oxide for its pains. And that ligand will occupy just those receptors and turn on or turn off, for example, a G protein, and activate or deactivate one neuronal pathway. Now, this sounds to me like the deep brain stimulation of the future. Obviously, how you're going to introduce transgenes into the human brain, but probably if you strip down viruses and make them, non-dangerous, you could probably do that. And you're only going to operate once probably, and it's no different from deep brain stimulation, which we know is going to have some impact in psychiatry. It's working to some extent in intractable depression, OCD trials.
OCD
very interesting,
certainly, yes, yeah.
So these are the kind of things of the future that I think we have to hold out for.
And the future of the BAP, I mean, currently we're very healthy, but
we are. Well, we're just having this
future?
wonderful meeting in Cambridge. I'm so excited that it's come back to Cambridge for this 40 years. We have
very appropriate.
over 500 people, lots of young people who I've met before. I think, this looks very rosy. I think we're going to have to change because of the way we're funded previously, but other major societies like ACNP and ECNP seem to have ridden over this problem of funding from drug companies. So I think we're just going to have to look for other sources of funding to take things forward, but I suspect we'll survive.
Well, Professor Trevor Robbins, thank you very much.
Thank you, Alan.